Okay. All right, Grady. This is the regular class section on Wednesday. We were just playing around for the photosynthesis lab. Next week, we've decided that we just needed to fine-tune the solution we were using, and instead of pulling the plunger in, you gotta pull it out in order to suck the air out of the leaf. So anyway, I'm sure one of your buddies here in class will fill you in on the technique before next week. Like I said, we won't do that lab till next Wednesday. Okay. But, however, I want to kind of review some pictures of the information we talked about yesterday. I'll start kind of easy here. These are the stages of mitosis in real cells. By the way, you should already know if this is a plant or animal cell. How do you know? Plant. How do you know?
Cell plate.
Yep, you see the formation of a cell plate over here. Okay, very good, guys. The phases are the same, though. Again, a couple things to remember about mitosis before you get into details is mitosis is the process where you are going to make genetically identical cells in the end with the same number of chromosomes. So if the parent cell had 46 chromosomes, you're going to make cells with 46 chromosomes. If the parent cell had 12 chromosomes, you're going to make a cell with 12 chromosomes. So this is the technique or the process we're talking about today. I like showing real life pictures because it kind of puts everything real. We did see a kind of a nice movie yesterday, but that's animated, which is kind of nice. Know the major things that happen at each step. Prophase, you still see that the chromosomes have not quite started to condense fully yet, although that's starting to begin. So the chromosomes start condensing, or I should say the chromatin starts condensing into chromosomes, if you want to use the right language. The nucleolus, even though you can kind of see it here, it does disappear by the end of prophase. You still do have a nuclear envelope at this phase, but that will also quickly go away here within the next step or two. Prometaphase, you actually can definitely see those chromosomes more visible now as packaged units. Remember, the whole point of mitosis is to just separate the genetic material, and it makes a lot of sense that genetic material condense first so that it can be more easily broken up into two places. But remember, at this point, you already have duplicated your genetic material. You did that a long time ago in interphase, in the S phase. So here you're just condensing those chromosomes of which you have copies. Metaphase, all those chromosomes are going to line up in the middle of metaphase. That's an easy way to remember it. Anaphase, those chromosomes will move away from each other in anaphase. Remember, even though you can't see the pictures here of the mitotic spindle, it's there. That's basically the equipment that's being used to push and pull these chromosomes from one location to another. And then, yes, you have the end of telophase. You still don't have exactly two cells yet. That's cytokinesis. Cytokinesis is actually when the cytoplasm divides and you actually have two fully formed cells at that point. So some real life pictures of that. I'm going to back up here and show a couple other pictures. They actually show, they show some real life pictures of all the things we've been talking about. Yeah, it's kind of neat to go in your book and see actual pictures of the spindle fibers and the chromosomes. Here's a nice close-up of what a kinetochore looks like. Remember, this is the chromosome, one of the sister chromatids, and you actually see the attachment point for that mitotic spindle. So it's kind of neat to see these. They do talk about this experiment in your book. Briefly, I'll explain what they actually... it's nice to be able to look at an experiment in your book, and you should be able to tell what's going on. That's going to be, the AP test is going to like you to know that. They'll probably throw a lot of experiments at you and expect you to interpret them. What this particular experiment is they were wanting to determine exactly how the spindle pulls the chromosomes away. In other words, is it like a rubber band and like tugging it? I mean, how basically do these the spindle shorten? They at first thought that it shortened from where it originated. So in other words, you have maybe at this end, the microtubules are kind of disintegrating. However, from this experiment, they kind of labeled a certain, like at the middle of the mitotic spindle, they kind of labeled it. And what they found out is that as the chromosomes get pulled away from each other, like an anaphase, as the chromosomes are moving away, the mitotic spindle is actually kind of disintegrating from the point of attachment of the chromosome. In other words, it gets smaller from this end, not this end.
So then how exactly does it get all the way over there then, or doesn't it need to get all the way over?
It does get all the way over. They haven't showed the whole way.
Oh.
They basically what will happen is disintegrate, and then it'll probably, stop at the very end over there where the centrioles are at. So again, not terribly important, but it's in your book, and I think it's kind of interesting to be able to look at a picture like this and interpret the data. We talked about that one of the differences in animal and plant cells is this cleavage is found in animal cells. This is cytokinesis now. It's a little different whether you're a plant cell or an animal cell. In animal cells, there's something that cleavage furrows form that actually split the two cells. In plants, cell plate. And I think that as far as, the last thing I was talking about yesterday before I got the bell rang was that prokaryotes actually divide, they actually reproduce by, see, I'm not going to call it mitosis. It's something called binary fission. It is separate from mitosis, but they talk about it in this chapter because, well, you're talking about cell division for the most part in this chapter. Mitosis is just a small part of that. So they talk about basically this prokaryote and the way it divides. And the last thing I think before the bell rang, we said, okay, it's kind of like, bacterial chromosomes are circular. So that big purple thing there is, even though it's huge and like condensed and folded on each other, it's literally one circular chromosome. Think of like a rubber band that's kind of like squished together. So you have a circular chromosome, and then you just kind of you fold it up like this and squeeze it into a cell. It's going to look like that purple thing right there. However, it's still circular. And what ends up happening is it's a really easy process in bacteria. They do not undergo mitosis because they don't have linear chromosomes. Literally at a certain point, okay, at a certain point called the origin of replication, and I think that's one of the vocab books, words in your book, the chromosome's literally going to kind of split in one area. It's almost like if you took this rubber band and you took a pair of scissors and you kind of cut it. Okay, I'm not going to be able to do this real well with this. But basically what happens in this is if I could keep pulling this, and it's not going to pull exactly this way, it's basically there's going to be an origin of replication. The chromosome's going to kind of bubble up. It is going to copy itself. And when it's all said and done, you'll have the identical chromosome, and you'll have two identical chromosomes. And they each literally just kind of, for reasons they do not know yet, the chromosomes just kind of collect on both sides, on either side. The book was even quick to mention the details of this process are not known yet, which I find kind of interesting. Binary fission is what this thing is called.
Was that what was happening at the beginning of the video the other day, or is that something completely different?
Totally different, because that was mitosis. Okay? That was mitosis where you talked about linear chromosomes making a copy of themselves. This picture is similar in the fact that the chromosome is making a copy of itself, but it copies itself a little different. It copies itself in like a bubble, kind of forming from a circle kind of deal. Okay. All right, what I want to talk about today, I'm going to plug along. I'll actually show you the picture first because it's kind of worth it, and then I'll kind of give you a couple of notes about it. Where in the world or how in the world did mitosis evolve? Remember, evolution's a real big part of this course. In other words, boy, I don't know about you guys, but mitosis seems pretty complicated to me with all the steps. And so the next thing is like, okay, well, how in the world did mitosis even get to where it is? It's such a complex process. they have this picture in your book, and they talk about, this is actually really interesting. Mitosis is not identical in all organisms. Some organisms have kind of variations of mitosis. Okay. The bacterial cell replication is probably the most basic. That's binary fission. All you're doing is basically making a copy, splitting the cell in two. Nothing complicated about it at all. That's not even considered to be mitosis at all anyway, but that's meant to be probably one of the earliest forms of cell replication in ancient cells. Dinoflagellates, that's just by the way, that's a protist. Okay, it's a type of protist. And if you look at how those actually divide, it's a little different than the way most eukaryotes divide. Dinoflagellates have a little variation. By the way, one big thing you can see right away is that the nucleus doesn't go away. Remember, in mitosis and the ones we've been talking about, the nucleus goes away. It disintegrates for a time. Doesn't happen in all organisms. Some organisms actually keep the nucleus. Also in dinoflagellates, the chromosomes are actually attached to the outside of that nuclear envelope. In other words, mitotic spindles aren't even doing anything. Now you do see some kind of remnants of a mitotic spindle there. They're calling them microtubules. The function they have in these cells is literally to kind of pull the cell apart, to kind of elongate the cell. It has nothing to do with moving the chromosomes at all. Okay, but there are some kind of similarities to some degree. Diatoms and some yeast. Diatoms are another protist, and then some yeast do this. Now, in these critters, you can kind of see that mitotic spindle is kind of it's very rudimentary, and it is attaching to the chromosomes, but it's not nearly as complex as what you see in most eukaryotes. However, in diatoms and some yeast, you're still not getting rid of that nucleus. Okay, you're still not getting rid of that nucleus. It's not dissolving. It's not degrading and then coming back. But what's my point of this whole picture? This is kind of an important concept because evolution is a huge part of this course. They're showing you in this picture that there's actually organisms that exist today that provide evidence for the evolution of mitosis. The fact that you didn't just start with bacteria and right away mitosis came into play. There are actually forms of evidence to show that it was a step-by-step process occurring over many of years. And there's still organisms that are around today that have that kind of, the kind of the rudimentary form of mitosis there. Kind of neat. It's kind of neat. All right, so let me flip on over to the overhead and we'll plug along this chapter a little bit and see how far we can get. Oh, there it is. Is today the second?
Oh,
or is it the third? Today's the third.
Yeah, faster. Good thing. I would have misled you.
Okay, so be sure you guys look at those pictures in the book. I think it's even titled The Evolution of Mitosis. Again, since that's one of the key parts of biology, the key fundamental pillars in this class, those would probably be where they pull some of the questions from. In a nutshell, I'm not going to draw all those pictures out, but I'm going to kind of give you some of the highlights. Bacteria, remember, they perform the process of binary fission, and the point of those pictures is to show you, to give you examples of how they believe mitosis evolved from binary fission. Binary fission, it's believed that bacteria were the earliest life forms anyway, and it makes sense that they probably replicated very similar to the same way they do now by binary fission. Oh, go ahead.
Is that why, bacteria hasn't evolved from binary fission because they don't need to? Is that why?
Correct. And actually, there's actually advantages of binary fission because it's so quick. And so a lot of bacteria have not exactly, it's a great method in that sense. Now, remember what I told you yesterday, though, bacteria do have the opportunity to exchange genetic material and change over time. So it's not like even though the process of binary fission itself is making an exact copy of that bacteria, that's not to say that before this binary fission process happened that their DNA had kind of mutated prior to that. The process of binary fission itself is quick and dirty. Again, this is why hospitals have to constantly deal with staph infections, which are bacterial infections and everything else, because this mode of replication is so fast.
Wouldn't that mean that once a bacteria is mutated, like you're never going to have the old bacteria again?
Not necessarily, no. That's why, new strains of antibiotics constantly need to be made and evolve and changed over time, because once an antibiotic comes out to treat a certain strain of bacteria, that bacteria more or less changes, and so they have to, and the antibiotics won't work on that one anymore. So yeah.
But like I'm saying, like those ones that wasn't working on anymore, like they won't ever come through again. Like how could the chances of it coming through again?
That I don't know. I think it gets a little more complex when you start talking about that, because who knows if that bacteria had a chance to go off somewhere else as well. I mean, that's kind of a complicated question. That's good though. Kind of some key points to kind of remember when looking at those pictures. So the nuclear membrane doesn't degrade. I mean, this is just some of the highlights I'm only writing down. So when you look at those pictures in the book, you can kind of remember, oh yeah, like the dinoflagellates and the yeast, their nuclear membranes actually don't degrade. Another thing is the chromosomes actually attached to the nuclear membrane. That's different from today. But it's definitely different from bacteria as well. Okay. So, I mean, just a couple of highlights. Those are definitely worthy pictures of going over and understanding. Okay. Questions on kind of those pictures? It's kind of interesting to understand how certain complex structures and certain complex processes have evolved, and this is kind of some of the evidence that they have for that. Totally shifting gears here. If there's no questions,
should we know? Like, do you think the AP test will want us to know like those?
I actually think you're more likely to see them give you an experimental setup and show you pictures like you saw and be able to identify some of the key differences between mitosis and most eukaryotes versus the prior ones. But I would be surprised if they made you memorize that on your own off the top of your head. But they may show you, they say, look at these pictures and provide some evidence of the way that mitosis is, that's what I can see them doing. All right. How the cell cycle is controlled. Okay. A cell has to know when it's time to divide. It's a complicated process. That's what the next part of this chapter is about. In other words, if you're a cell, how in the world are you going to get signaled to that it's time to divide? And it's actually, believe it or not, actually, maybe easier than you think. But they refer to this kind of checkpoints word. There are certain points where stop-and-go signals are going to regulate the cycle, okay? Control points where stop-and-go signals regulate the cycle. In your book, they kind of compare it to a washing machine. They say, okay, think about like the rinse cycle versus the spin cycle versus all these different cycles in a washing machine, and they say that one doesn't start before the other one is finished, and they kind of compare that to the cell cycle, saying, well, a cell isn't going to start the S phase until so-and-so is done, and it's going to move on to the S phase, and the cell isn't going to move on into the G2 phase until these things have done in the S phase. In other words, there are actual checkpoints in a cell cycle, just like there's a spin cycle and a rinse cycle in a washing machine, where a cell has to have certain things lined up and ready to go before it can move on. That's what these checkpoints are. There are actually they say there's 3 major checkpoints. There's more, but there's 3 major checkpoints that occur at the G1, G2, and the M phase of a cell cycle.
What is the M?
M is mitosis, by the way. That's what I was about to say. When you see the M phase, it's actually referring to mitosis. And I'll actually kind of put that in. When they refer to the M phase, that is mitosis.
If they get to a checkpoint and they can't go, do they just stop there, or do they change it so they can't go?
This is exactly what happens. Basically, if they get the signal, they'll move on to the next one. If there is no signal, that's exactly what you were going to say, they actually go to this other phase called a GO phase. I don't know if it's given any other name. It's just kind of a G0 phase, a G0 phase. And it's basically what that is a non-dividing state. That's kind of like a cell saying, okay, it's obviously not time to divide yet. I'm just going to continue doing my things in interphase that I'm doing. It's kind of like a halting point. So that answers your question. Yes. If the signals aren't received, then it's basically going to stay there in something called the G0 phase.
So if it does, like, let's say it gets to G2 and then it says stop, you can't go any farther. Is it going to stay in G2 and then later on it will go into mitosis, or will it go back to the beginning? Like, will it go?
Oh, no, it'll probably stay the G2 phase until it receives the signals. Yep. It won't go backwards. Yep. Good question. Good question. Excellent. But it may halt, and when it kind of halts, they kind of call it the G0 phase. Okay. Okay, well then let's kind of talk a little more in particular about that. They actually, you might have talked about this in basic bio. I think I remember hearing Ms. Sin talk about it from next door. When it comes to regulatory molecules of the cell cycle, there's really one really big one. Let's see if you guys have heard of this before. I don't talk about this in my biology class, or I haven't, but I think I heard her talking about it. Let's see if it rings a bell. There's something called cyclin, which I'll kind of give you a brief definition. It's a protein. I'm just going to call it, it's a protein, hence the word, that cycles in a cell. In other words, this is a protein in a cell that at times is really high in concentration and at times is really low in concentration. So it cycles. It goes high, it goes low. It goes high, it goes low. Okay? That's natural. Another thing about this cyclin is that basically it starts being made in the S phase. So when a cell reaches the S phase, this is where cyclin starts producing itself. Okay. It accumulates in the cell. I imagine since it's a protein, it's just made on a ribosome and accumulates in the cell. Yep. Since it's like every other protein, it's made up by a ribosome and just kind of accumulates. Okay. Okay. There's another molecule that works hand in hand with cyclin called cyclin-dependent, because they work together, cyclin-dependent, you've heard this word, kinases. Okay, what kind of molecule is this? It's an enzyme. Very good. By the way, it's a kinase, to remember what enzymes that our kinases do. That's right. These are enzymes, and I'll write it again. If something is a kinase, it's an enzyme that adds a phosphate to a molecule. Okay? Okay. All right. So here's the deal. By the way, these cyclin-dependent kinases, these enzymes are all over as well. These are basically enzymes like every other enzyme is capable of recycling itself. So it basically is always kind of present. Okay, but cyclin-dependent kinases, when given the chance to hook up to cyclin, will. So when cyclin starts accumulating and accumulating in a cell, these cyclin-dependent kinases are going to start hooking up to that cyclin. Why? Because when a cyclin-dependent kinase is not hooked up to a cyclin, it's inactive. In other words, a cyclin-dependent kinase is inactive when it's not hooked up with cyclin, but it becomes active when it does hook up with cyclin. And as the cyclin is kind of producing itself, it's making more of itself. These cyclin-dependent kinases that were just roaming around there inactive are now hooking up and becoming active. And a lot of them are becoming active because more and more cyclin is accumulating. Okay. I'm going to kind of say, when they combine, they form something called MPF, which is called, technically the name for it is a maturation, like mature, maturation promoting factor. That's what it technically stands for, maturation promoting factor. But an easier way to remember this is by what is calling it an M phase promoting factor. Here's why. So I would know MPF as a M phase promoting factor. When these two molecules hook up, cyclin and the cyclin-dependent kinases, they form this MPF. They literally will change into that molecule, MPF, by having the two combined. The more MPF that accumulates, that's the signal to start mitosis. Okay.
I'm not understanding which one it stands for, the M phase.
Either one. Technically, it's called the maturation promoting factor, but I'll be honest. I think an easier way to remember it, and one that I don't care that you use on a test or whatever, is M phase promoting factor, because that implies that this complex, this MPF, which is basically these two molecules held together, is basically what's going to push you to mitosis. This is how mitosis takes place, how it starts, how it progresses, and how it ends is with the appearance of this MPF. Okay, a little more detail, and I'll show you a picture, and it'll all make sense. MPF, by the way, a couple more notes. What this thing does, remember, this is like the active form of this kinase. So even though this was a cyclin-dependent kinase, it didn't do anything. It was in an inactive form. But now that it's MPF, it's an active form. So now it can go on and phosphorylate a bunch of molecules. It phosphorylates molecules, causing mitosis to begin and progress. Again, progress. Phosphorylates molecules, causing mitosis to begin and progress. Let me give you an example. Remember, one of the steps of mitosis is that nuclear envelope degrades. How in the world does that nuclear envelope disappear? Well, this MPF molecule will phosphorylate the molecules in the nuclear envelope, and it'll signal it to degrade. Okay? It basically, in other words, MPF is the molecule that is going to make things happen in mitosis. Where does MPF come from? It's basically the resulting molecule when cyclin and cyclin-dependent kinases hook up and do business together. Yeah, go ahead.
MPF is what signals the cell to go from G2 to mitosis?
Yes.
Oh, and I guess not necessarily signals, but what causes the reaction? What causes the cell to go from G1 to S and S to G2? Or even from, yeah.
You know, they only talk about this being the big one to go from G2 into mitosis. My guess is that there's other molecules that are doing different signals, but they only talk about this one. Yeah, this is the only one that I think I know any detail about, is the one that'll take you from G2. And I should probably say this. This whole process takes you from the G phase 2 in through mitosis. This is the one, I don't know if this is because they know more about this checkpoint than any of the others, but they know that there are checkpoints there. But this is basically the checkpoint that is they know a lot about, and it's the one that's talked about, cyclins and cyclin-dependent kinases. By the way, I would almost really make a note. This is like AP exam question, big time, understanding it, and I'll show you a picture here. This is one of those things I can see them talking about.
So how come like the S phase doesn't really come in with like the whole checkpoint thing? Like it's in between the two.
It's in between, and you know what? There must be other molecules that regulate the passing from the G1 to the S phase.
So it has nothing to do with the checkpoint stuff. The S phase.
I don't believe so. If there's not a checkpoint there, which I don't think there is, then it doesn't seem to be a major checkpoint. There's probably, it's considered like a minor checkpoint, the details of which I don't know. Okay. This is definitely one of those major checkpoints that we talked about, that they are important. Okay, let me show you a picture. This might make a little sense when we kind of look at a picture. Oops, this is kind of low. Sorry about that. I didn't realize that. There we go. Okay. They do talk about that experiment in your book. I'm not terribly concerned in going over that one. Okay, these are kind of like those checkpoints we were talking about. There are multiple checkpoints throughout. Now the one that I was talking about with the cyclins and the cyclin-dependent kinases, that's kind of this one right here. There's other checkpoints. And these are the major ones, but there are other ones that, you know, details of which you don't need to know. But again, kind of think of it like a washing machine cycle, literally. You know, you go and you start, you fill up with water. Then it kind of flips over, then you go the agitation. Then you know what I'm saying. They actually, it's a very good analogy. When they receive this signal, it's going to go. If they don't receive the signal, it'll go into this G0 phase, which is just kind of like an inactive form, or just they call it a non-dividing state. Basically, this cell is not going to divide.
So it kind of just extends whatever phase.
It does. It extends the phase. Let me realistically put cells on this. You know, not all cells in your body, for example, replicate at the same rate. Your liver cells will replicate once a year. Your skin cells will replicate once a day. In other words, these cells don't go through a cell cycle at the same rate. So a lot of our cells in our human body, with the exception of our skin cells, are kind of at this G0 phase. They're not actively dividing cells. The actual division process itself takes such a quick time. A cell spends most of its time in interphase, if you remember. So interphase is basically kind of like G0 would be considered kind of a part of interphase. Yeah.
So is it not stopped because there's something wrong, or is it stopped because there's something wrong, or is it stopped because it's not needed yet?
It's not needed yet. It's not needed. It's not like something's bad. It's literally it hasn't received the signal to go yet, and that's not necessarily a bad thing at all. Literally, the signal has not been received. It doesn't need. It can keep doing. Remember, even in an interphase, a cell is actually a very productive cell, making its proteins, making its carbohydrates, doing the things that the cell needs. It doesn't have to actually absolutely divide to be healthy all the time. So. Now, let's stare at this for a while. This is really important stuff right here. This has AP exam, I think, written all over it. If I had to pick a few of the topics that I see this. By the way, the reason I also bring that up is that one of the labs in your AP book that we're going to be doing is a mitosis lab, meiosis lab. And they again put this picture up there in your lab notebook. So I said mine. Okay. Not only is it covered in material in the book, they also covered it in the AP book here. I'm thinking you are going to see this at some point on that AP exam. So let's make sense of this one, definitely. Let's look at the bottom first. I think it's easier almost to look at the bottom. Let me tell you what's going on. Let's start with the S phase. S phase, something important starts happening. This protein called cyclin is starting to accumulate. They represent that by that purple thing. Okay. These cyclin-dependent kinases, they also call CDKs. Okay, CDKs, cyclin-dependent kinases, they're always there, by the way. You can even see that here. These things are like recyclable molecules. They're enzymes. Enzymes mostly are recyclable. As long as the temperature and pH are happy, those things are going to live forever. Okay? So you have this cyclin-dependent kinase that when this cyclin starts to accumulate, they're going to hook up together to form that promoting factor, mitosis promoting factor. When this accumulates is basically the signal to start mitosis. When enough of that accumulates, it's like ding, 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 mitosis. That stuff's going to keep on kind of accumulating for a while, we'll see. But towards the end of mitosis, actually anaphase to be exact, what's going to start happening when anaphase goes on is this cyclin's going to start degrading. Okay, that's not a bad thing. It's just going to degrade. Remember, it's a protein. Your cells can make more. But it's going to start degrading, but that will basically switch off. That basically like a light switch switching off mitosis, saying it's coming to an end, mitosis, and that cytokinesis needs to happen. Okay, very, very important to kind of understand this kind of whole setup. These things work hand in hand. When this stuff starts accumulating, it'll bind to this stuff, which is already there. They will form this kind of unit, this molecule, MPF, which is going to signal mitosis to go. I went into a little detail in your notes and said what exactly this thing does. It's an active enzyme, more or less, which is going to phosphorylate molecules, getting them to do things. For example, it'll phosphorylate those molecules on the nuclear envelope, getting it to degrade. It'll do other things in the mitosis process as well, but I'm not worried you know those super details. Just other things that take place in mitosis are a result of this guy right here. Toward the end of mitosis, during anaphase, cyclin will start degrading. That shuts off the function of that molecule. That basically says to the cell, all right, time for cytokinesis. Bam. That's when you get your two cells and the whole thing starts all over again within those cells. Yeah?
If the cyclin didn't degrade and it maintained the form of MPF, would mitosis happen again? Would the cells divide again?
That is cancer. Yes. And that's when problems happen. That's one reason you can get cancer. There's multiple reasons. But yes. Very good. Good. Hey, look at above. Okay? Hey, I had to stare at this thing for a while above because I think I was reading it too detailed. That graph above is kind of saying the same thing below. It's taking let's take them one at a time. Cyclin. Cyclin, like the word, cyclin, cycles. You can see from that line that you actually have cyclin being high in some phases of the cell cycle and low in others. And you can see it peaking. It peaks most pretty much around this phase. You have more of these purple things at this point than at any other point just before anaphase. You have a lot of them. That's why the peak is the highest for cyclin then. But in anaphase, basically, it's going to just plummet because all of those cyclins are just going to degrade pretty much in anaphase. So whoosh, they're all going to go down in concentration. So you can see the purple line, cyclin concentration up and down, peaking at anaphase. There you go. The orange line is representing the activity of this guy right here. Remember, this is the guy that's phosphorylating all these molecules. And this is basically telling you that during mitosis is when this molecule is active and doing business. It's phosphorylating molecules like crazy. So it obviously peaks at mitosis and only mitosis. That's the only time it's formed. Okay, so that's what that graph above is telling you there. In other words, cyclin concentration and MPF activity kind of go hand in hand, but that makes sense. Because the only way MPF is going to do some activity is if enough cyclin is around for it to form. How we doing? This is kind of complicated. Raise your hand if you kind of sort of get it. Okay? Raise your hand if you are clueless. Raise your hand if you're like, okay, I think I get this. All right. You guys all right with this? Is there anybody? Okay. I like this. and I'm glad we're taping this for Grady. So Grady, you know, come see me with questions on this one because this is actually kind of important stuff here. Like I said, when I saw it in your lab manual, I kind of instantly said, okay, it's going to appear somewhere on that AP test. So it's a good stopping point, I'll tell you that much, rather than going on. Unless there's any other questions. All right. I'll kind of shut down here and